Good morning, good afternoon to everyone from all parts of the globe that you are joining us here today for our seminar, Use of PNI Microfluidics to Manufacture GMP Quality siRNA Nanoparticles. The speaker of this webinar is Dr. David Evans, Chief Scientific Officer of Sirnaomics. David Evans received his scientific training at Imperial College London and has over 15 years of experience in the RNAi therapeutics field. Prior to founding Sirnaomics, he worked at Serono and at Millennium Pharmaceuticals. The topic of the presentation is, as you see here, and David leads the scientific team as the Chief Scientific Officer at Sirnaomics. I'd also like to give a shout out for the next webinar we have planned for you, and this is June 25th at 7:30 Pacific time. And this will be provided by Dr. Marcel Bailey, a renowned scientist in the field of lipid-based nanomedicines, and really the challenges that he has come across in his 35 years of research. And again, this is a webinar, the next webinar, which again will be very exciting. So just quickly, who is PNI? You may all know us, but we are a high growth technology company enabling the accelerated production of transformative medicines that impact human well-being. We are a scientific team in Vancouver working on technologies to meet market needs, and a multidisciplinary professional team across the globe to support you in bringing these solutions and providing you the technical support that you need. PNI provides complete solutions, and this is including our systems, our reagents, and services across the drug discovery pathway. What David will touch on today is our preclinical, late preclinical, and clinical systems, which is the BLAZE and the GMP system. Additionally, as you can see, PNI has novel lipid formulations for clinical licensing and for research use. And we also have an expert scientific team in our Vancouver facilities that can enable you with taking your formulation all the way from proof of concept to GMP manufacturing. So with that, I'd like to turn it over to David for his presentation. Thank you.
So thanks to Pamela and to PNI for inviting me to speak today. I'm going to be telling you about our experience in using PNI Microfluidics to manufacture GMP quality siRNA nanoparticles. I'd like to thank PNI for the opportunity to present this work and to all of you for attending. So Sirnaomics, for those of you who don't know the company, is a global clinical stage RNAi company. We have footprints, headquarters and main R&D offices are in Washington, just outside Washington, DC in Gaithersburg. And we also have labs in Suzhou and Guangzhou in China. And we have offices in Boston and Hong Kong. So we're a pure play RNAi therapeutic company focusing on fibrotic diseases and cancer. And we've got an enriched pipeline in clinical and preclinical development, which I'm going to share some data with you today. So in terms of technologies, Sirnaomics has basically built out the capabilities in all aspects of RNAi discovery through development and into the clinic. So we have the ability to do target selection, oligonucleotide therapeutic drug selection, in vitro studies. We have a novel delivery vehicle, which I'll tell you about in a little bit, but it allows delivery of 2 siRNAs at the same time. So we leverage that opportunity. And we have capabilities in vivo studies, CMC formulation, and clinical phase 1 and 2 clinical operations currently in place. So in terms of our delivery technology, we use a histidine lysine polypeptide. It's a branched polypeptide shown in the figure on the top left, which is 4 branches, and the lysine moieties in the histidine lysine polymer form a ionic bond with the phosphates in the siRNA. The histidines help with delivery of the siRNA into the cell. So when we form these polymers with the siRNA, we get self-assembled nanoparticles. When we deliver these, we've done intradermal, intratumoral, and IV administration of these nanoparticles. And when we deliver these into these delivery mechanisms, this nanoparticle will interact with the cells, gets taken up into the cells, where the histidines play a role in releasing the siRNA. So the pKa of the histidines is about 6.3, and as the endosome acidifies, then the histidines eventually become positively charged, and that causes lysis of the membranes and release of the siRNA into the cytoplasm. So the unique properties of HKP are that it will deliver and target to the tumor microenvironment. So we benefit from the EPR effect, enhanced permeability and retention effect, with the size of our nanoparticles and the nature of these. And I'll be addressing that more as we go through, and I detail our results from the GMP production of a batch of our nanoparticles. So when we deliver IV, we deliver to cells beyond the hepatocytes, which I'll detail in a little bit as well. The advantage, as I mentioned earlier, is that the HKP can carry 2 siRNAs in the same nanoparticle. So the rationale for including a second siRNA. So our drug product targets 2 genes at a time, is designated as a single agent by the US FDA. So it reduces complexity and cost of preclinical and clinical trials by having a single agent rather than 2 separate agents needing to be co-administered. By silencing 2 targets using the same nanoparticle, we can focus potency by affecting 2 targets in the same cell with additive synergistic effect. This helps reduce chances for cells to escape therapeutic pressure through target mutations or changing levels of a single protein. So in some cases, this is very important. Identification of targets, we typically will validate our siRNAs against a specific target and then do rational testing of combinations of validated siRNAs to identify synergism in different targets. And we also spend a lot of time, because our focus is in fibrosis and oncology, we also spend a lot of time looking at oncology standard of care agents to identify synergistic response with our siRNAs that way as well. So this is an example of 2 siRNAs that would probably not pass much in the way of interest in terms of delivery for treating cancer. This is a head and neck cancer, 2-cell models, FADU cells and SCC25 cells. And I can't tell you the nature of the siRNAs, but I show siRNA number 1 and siRNA number 2 in pink and purple, and the combination is in blue. And you can see dramatic increase in performance when the 2 are combined in FADU cells with a combination IC50 of 0.8 nanomolar. And in the squamous cell carcinoma 25 cells, we see that the combination produces an IC50, and this is in viability of cells, not knockdown, of 0.08 nanomolar. So you can get very great synergy from combining these 2 siRNAs. We were fortunate in the fact that these 2 siRNAs are also functional in bladder cancer and again show a combination effect that's synergistic. So siRNA number 1 and number 2 in SW780 cells in the top figure, combination IC50 is 0.14 nanomolar, and in UMUC3 cells, another bladder cancer cell line, we see IC50 of 0.8 nanomolar in the combination. So we've leveraged this ability to target two siRNAs to two different targets at the same time. And we've got two lead candidates in clinical development, STP705 and STP707. The difference is STP705 is for local administration, STP707 is for systemic administration. Both products are HKP delivering TGF-beta1 and COX-2 siRNAs. The STP705 is currently in phase 2 studies in non-melanoma skin cancer. And we reported recently that we saw tremendous efficacy of this product in clearing non-melanoma skin cancer in patients in a dose-ranging study at the medium dose that we've tested so far. So that's looking very promising. So STP707, though, is delivered systemically and is a slightly different formulation in the terms of the concentration of the product. And we need a GMP batch to perform required toxicity testing and clinical studies for this delivery vehicle. So in terms of fibrosis, TGF-beta1 and COX-2 have multiple roles in fibrosis and oncology. I'm going to show you some data initially in fibrosis. So, and all of the data I'm going to share with you today is focused on liver delivery. So you can understand the rationale for wanting to generate the GMP product for that indication. So TGF-beta ligands are potent drivers of extracellular matrix deposition and create a concentrated pool of pro-fibrotic factors within the tissue. When we deliver HKP siRNAs, this is a study done by Taiwan University where they used fluorescently labeled siRNA formulated with HKP into nanoparticles, and they delivered 20 micrograms in 200 microliters per animal, which is about 1 mg per kg. And you can see in the lower figure the uptake into various cell types at 1 hour, 4 hours, and 24 hours. And the red bar is Kupffer cells, the blue is dendritic cells, the yellow is liver sinusoidal endothelial cells, and the gray is hepatocytes. So you can see that we get very rapid uptake into Kupffer cells and hepatocytes at 1 hour post-treatment. And the hepatocytes retain this product much greater. At 4 hours and 24 hours, we still see the fluorescent siRNA in those cells, as well as in Kupffer cells. Stellate cells were difficult to monitor because they represent less than 1% of the total liver cells. And so in the flow cytometry experiment performed here, that was very difficult to detect stellate cells. So this slide shows liver delivery to primary stellate cells, the human primary stellate cells. So on the left, we show that HKP-mediated fluorescent uptake of siRNA was greater than lipofectamine RNAi max. And on the right, we show that at 72 hours post-transfection, HKP with a cell death siRNA caused greater cell killing than lipofectamine RNAi max at the same concentration. So we demonstrated that we could deliver to the human primary stellate cells. So with this, we felt that we could obviously treat liver disease with the nanoparticle. And so we built an animal model using carbon tetrachloride induction in mice, BALB/c mice, 4 weeks of 2 times per week injection of carbon tetrachloride induces fibrosis in these animals. Then we followed that with STP707 treatment for 3 weeks, 2 times per week. Then we harvest the tissue, and we did histological observations of the samples. So this screen shows the effects of these treatments. So the blank at the top is untreated animals. The model shows collagen deposition in the white spots. And the model non-silencing treated siRNA shows still some white spots present in the liver. And the two lower sets of panels show results for 0.5 mg per kg and 1 mg per kg STP707 delivered into the liver. And you can see that in these treated animals, we see very clean response, no collagen deposition. So suggesting that the product is efficacious. So in oncology, elevated TGF-beta levels produced within the tumor site have been associated with a reduction in the ability for T cells to infiltrate into the tumor. The TGF pathway exerts most of its pro-tumoral effects by remodeling the tumor microenvironment. And COX-2 is also suspected to promote angiogenesis, resistance to apoptosis, and also may augment the conversion of active T cells to Tregs. So what we wanted to see was if we could reduce the amount of TGF-beta1 in and around the tumor, could we observe better uptake of CD4, CD8 positive T cells into the tumor site? And seminal papers by Torriello and Mariatherson in 2018 in Nature showed that TGF-beta acts as a wall to prevent access of T cells into the tumor sites. So we wanted to see if we could convert a cold tumor with high TGF-beta levels and poor response to immuno-oncology therapy such as PD-L1 monoclonal antibody into a hot tumor where TGF-beta levels are lower and you have a sensitized response to immuno-oncology therapy. So for those of you who are not familiar with immune checkpoint inhibitors, PD-1 exists on T cells. PD-L1 is a target receptor present on tumor cells. And when these two are combined or bound, the tumor cell is sending a signal to the T cell, don't eat me. So there's no killing of the tumor cell. So over the past several years, there's been tremendous interest in developing antibodies that will block this interaction between PD-L1 and PD-1. And several PD-L1 antibodies shown here, durvalumab and atezolizumab, have been developed, and several anti-PD-1 antibodies shown here, nivolumab, pembrolizumab as examples. So these antibodies bind to one or other of the targets and prevent their interaction. So this then allows the T cell to induce its tumor cell death and kill the tumor cell. But as I mentioned, with the TGF-beta levels upregulated, the question was whether the T cells can even penetrate to access the tumor site in the first instance and become antigenically primed because of the TGF-beta wall that's induced. So we wanted to examine this. So we used STP707 intravenously combined with PD-L1 monoclonal antibody to treat HCC in mice. So day 5, the mice are randomized. This is a HEPA-1/6 luciferase expressing cell line. So it's a syngeneic orthotopic model. So the cells are injected into the left lobe of the liver, and these are monitored using IVIS in vivo imaging system. So over time, with control treated, vehicle treated animals, the tumor grew very rapidly, as you can see here. The two bars shown at the top here are where we had to take last observation carried forward, because on the right-hand side in the Kaplan-Meier survival curve, you can see that we lost 50% of the animals throughout the dosing phase, shown in gray in both these images. So sorafenib is a gold standard for HCC treatment, so we examined that. An anti-PD-L1 monoclonal antibody alone, we examined that in combination with STP707. So STP707 alone is shown in green, and STP707 plus anti-PD-L1 monoclonal antibody is shown in the blue. So as I mentioned, we lost, so what we saw was that the tumor reduced so dramatically that there was no tumor left, essentially, after 22 days treatment with STP707 or STP707 with the anti-PD-L1 monoclonal antibody. So there didn't seem any point in trying to rescue the tumors and trying to elucidate whether the CD4, CD8 positive T cells had penetrated. So we let the experiment continue to see if there was any recurrence of the tumor resulting in death of the animals. And you can see that in sorafenib, we see loss of one animal, and in all the other groups, STP707 and anti-PD-L1, we didn't see any increase in cell death. So since we couldn't recover tumors from the previous study at 2 mg per kg, we decided to treat only 3 doses at 1 mg per kg and rescue the livers out of the animals and explore the size of the tumor and the CD4, CD8 positive T cell penetration into the tumor. So the figure shows the livers outlined in green, and the tumor here is in the vehicle treated is outlined in red. And the colored segments on the right-hand side figure show different layers that we would then monitor for uptake of CD4, CD8 positive T cells into the tumor. So after STP707 at 1 mg per kg and 3 doses, you see that there's a dramatic decrease in the tumor size. And that is also correlated with this increase in CD4 and CD8 positive T cell penetration into the tumor. If we monitor from the liver into the tumor at 50 micron increments and quantify the CD4, CD8 positive T cells from the images, you can see that in the control arm and anti-PD-L1 alone, we get a slight increase into the tumor. With STP707 plus anti-PD-L1, however, we see a dramatic increase in CD8 positive T cell penetration into the tumor. So this would suggest that this would have a therapeutic benefit in treating HCC. So with that, we've demonstrated that in fibrosis and in oncology, we have indications in the liver that would require us to be able to provide intravenous administration of our product. So now I'm going to talk you through the steps we took to generate GMP quality product. So we had acquired a benchtop instrument from Precision Nanosystems, and that came with a herringbone pattern mixer, and we had used it to formulate HKP with siRNAs. So HKP goes in one channel, siRNAs mixed together, the 2 siRNAs mixed together go in the other channel, and we generate the polypeptide nanoparticle out of the other end. So by varying flow rate, we can show that we can reduce the size of the particles as we increase the flow rate in this system. And the optimal flow rate for us was about 8 mils per minute, producing particles about 100 nanometers in diameter. And you can see the consistency in the polypeptide nanoparticle size from each of these steps. So that's all very well and good, but at 8 mils per minute, we'd never be able to produce enough GMP product to satisfy our needs. So we were fortunate that Precision Nanosystems was working on a new device, the NxGen mixer. This is a Dean Vortex mixer that uses centrifugal force to mix the fluids in the microfluidic chip. So similarly, HKP and siRNAs are introduced in the left-hand side, and the nanoparticles come out to the right-hand side of this device. The advantage of this device is it can flow at 0.2 to greater than 20 liters per hour, so that shouldn't be an impediment to the amount that we need to make for GMP production. And as before, we validated at PNI that increasing flow rates reduced the size of the nanoparticle, and sure enough, we saw that with a consistent PDI occurring after 75 mils per minute. And that's shown here, so in the tabular form. So at 8 mils per minute using this mixer, we saw very large particles, but by 75 mils per minute, the particle size had reduced to 111 nanometers, PDI was 0.25, and the zeta potential was around 38. At 120 mils per minute, we're seeing 87 nanometer particles, 0.22 PDI, and 34 millivolts zeta potential. So this was right on the money for what we were expecting. So the consistency of the product is shown here. So at low flow rates, up to 120 mils per minute, we see that the consistency improves. But at 120 mils per minute and 200 mils per minute, we see very consistent results. So we had performed those studies using 4 mg of siRNA as an input, and we'd used 90 mils per minute ostensibly as the flow rate, and we produced particles of 100 nanometers diameter with very consistent polydispersity. So we wanted to see if this scaled. So we used 60 mg of siRNA, same flow rate. We see very similar particle size, very similar PDI, and similar zeta potential. And the same is true at 200 mils per minute. We see 70 and 66 nanometer nanoparticles at the 2 different loadings, and 0.21 polydispersity index, and 34 and 35 zeta potential. So formulation using Microfluidic Microfluidic GMP production, this is a flow chart of the steps we took to validate the GMP production. So raw material dispensing and sterile filtration is performed up front. Microfluidic mixing is performed as the next step. We fill a 20-liter biobag, lyophilize the product, stop and crimp in vials, and do visual inspection. So the GMP quality APIs and excipients used were HKP from Chinese peptide company in China. HKP plus H is a variant of HKP. It's produced by PPL, Polypeptide Labs, and each of these was at 4 grams. STP705-1 is TGF-beta-1 siRNA produced by Nitto Denko. STP705-2 is COX-2 siRNA produced under GMP conditions at Nitto Denko, and each of these was at 1 gram. So our flow chart shows that we start with HKP bulk solution and RNA bulk solution in the 2 arms of the nano mixer, and up front, we perform 3 filtrations, 0.45, 0.22, and 0.22 micron filtration. The idea here is to create sterile product before mixing, and then collect in the biobag and measure the aseptic capabilities of the device afterwards. So this is the NxGen system. It's basically consists of the microfluidic device enclosed in a block of stainless steel shown on the right-hand side here. And it has 2 inlets and an outlet for connecting to the pumps. So the most important part about this slide is that the quality documentation meets CGMP requirements. So this was something we were working on in parallel with PNI as we were developing this process. And PNI was able to obtain CGMP status for the product. And it's a fully disposable fluid path. So our engineering batch was produced. It lyophilized exceptionally well. We see the results here on the left-hand side. And we rehydrated it in 3.2 mils of water and evaluated it for consistency. And you can see the average size, 99 nanometers, is right on the money for what we were wanting. The PDI is lower than we've seen previously at 0.16. Zeta potential was correct. The siRNA concentration was correct, and the pH was exactly the same as before. And we see the consistency of the peak size from the batch being measured in the zeta sizer. So then we moved to the GMP batch, and here we used 0.32 mg per vial. HKP siRNA ratio is 2.5 to 1, and the HKP lots are shown there along with the STP705 lots. And characterization of this product, again, we ended up with 102 nanometer particles before lyophilization, very consistent. PDI, very consistent. Zeta potential, siRNA concentration. And the color and clarity of the solution was opalescent with no particulates, which is very important for an IV administered product. pH was 5.91. After lyophilization and rehydration, the size went to 107 with a PDI similar to what we saw before. Zeta potential was exactly the same. siRNA concentration went up just a little bit. Same color clarity of solution, and the pH was identical. So next steps, STP707, we want to use this for in vivo toxicity testing in non-human primates for systemic administration, and IND filing for specific indications, as I mentioned in the research work that we've done previously. So in summary, we demonstrated that NxGen microfluidic technology from PNI can be successfully scaled to industrial levels in GMP manufacturing. The microfluidic system offers precise control of particle size and consistency in nanoparticle formation, which greatly accelerates process development from benchtop to products. And Sirnaomics has used the technology to manufacture 2 engineering batches and 1 GMP batch of STP707 drug candidate. The QC release test of the 3 batches met all of the specifications, including bioburden and sterility. So in terms of the timeline for producing this GMP quality product, we started in October 2018 with the purchase of the benchtop instrument and the herringbone pattern micromixer. We started speaking with PNI in June of 2019 about GMP production, and we performed formulation development and scale-up to anticipate the ability to use the system for GMP production. In November 2019, we signed a license and supply agreement with PNI, and in December, we performed tech transfer of formulation and GMP system to the clinical medical group. And in January 2020, we were able to run the engineering batch run. And April of this year, right before COVID hit, we were able to manufacture a GMP batch that's subsequently been validated for bioburden, et cetera. So proof of concept to GMP in 6 months from initiation. It's very quick. So PNI Microfluidics, key features for success. The NxGen mixing at higher flow rates mimics the benchtop mixing at R&D scale flow rates. So this allows confidence in development of processes at R&D scale and transfer to GMP production. Using NxGen GMP microfluidic system allows high flow rates, allowing multiple liters of manufacturing capability, minimizing the clean room time. And the consistency of the product at a given flow rate, limited size distribution, and reproducible zeta potential was excellent. So it met all of our criteria. And the QC release test of the 3 batches that I mentioned earlier met all the specifications, including bioburden and sterility. So with that, I'd like to acknowledge the following contributors. Professor James Mixon, University of Maryland, for the inventor of HKP for delivery. Crown Bio US and UK for doing the in vivo studies that I mentioned earlier. Precision Nanosystems, of course, for developing the products that we were able to use in today's seminar. And Berkshire Sterile Manufacturing, the contract manufacturing organization in Lee, Massachusetts. And within Sirnaomics, I'd like to specifically point out Stephen Long, Mark Lemaitre, Eric Roche, and Edward Wong in the CMC group, who were primarily responsible for enabling us to perform these experiments as efficiently as we were, bringing HKP and siRNA GMP quality products together and testing the NanoAssemblr for viability. So that's it. I apologize for the earlier problems with the presentation, but if you need more information, my email address is here. Please reach out and let me know.
Thank you, David. Thank you, David, so much.
So we have a number of questions. Unfortunately, we can't get to all of them. Okay, one of the questions would be here was, were there cryoprotectants in the formulation during lyophilization? I guess that's a question for David. Are you able to speak to that?
So Yeah, we've evaluated both adding cryoprotectants and without, and the data I presented was without, but we've also validated that we can add cryoprotectants and improve the lyophilization.
Okay. Another question is, how many grams of siRNA were produced in the GMP batch?
That's a good question. So I think, according to, I'd have to check on that, but I think it was the 2 grams that I mentioned in the earlier slide.
Okay, very good. Again, I think you spoke to this, but what is the final dosage form of the formulation? Is it liquid or a lyophilized solid?
So it's a lyophilized solid that can be dispensed, it can be re-administered with liquid for injection. So that's the aim of the product.
Very good. Okay, and a question here you may be able to answer. Do any modifications on amino acids affect encapsulation of siRNAs or their intracellular release?
So yeah, so the HKP plus H formulation is basically HKP with an extra H present in one arm of the polypeptide, and we see some change in efficacy between HKP and HKP plus H in terms of delivery, but not sufficient to concern us about the change in product.
Okay, very good. So I think those are the probably. I think that's all the time we have right now for questions. But I think we can certainly.
Lloyd, I have one question. So, David, I know the current particles, HKP particles they are making, go to the liver. But are you able, with the structure that you have with the HKP, do any targeting by, putting any modalities within your nanoparticles to get them to target to specific sites or organs?
So we're working on that at the moment. So yeah, that HKP being a peptide, it's very amenable to adding additional ligands onto it to help with the targeting delivery. And we're exploring various means to modify it to get us to different tissues. Primarily, our interest right now is in targeted delivery to pancreatic cancer and lung cancer.
But Lloyd, I don't know if you could just speak to just a second maybe about our GMP system.
Yeah, certainly. So as Padma kind of introduced in the beginning of the talk before David's again giving his presentation, PNI has a number of systems for manufacturing or producing nanoparticles of all different shapes and sizes. In this case, it's a siRNA-based nanoparticle for the presentation today, but as you know, we can also produce different kinds of nanoparticles, including peptide, seeing here, polymeric nanoparticles, lipid nanoparticles, liposomes, and emulsions. Focusing on lipid nanoparticles, we have worked with a number of clients that have adopted our NanoAssemblr technology to scale up these promising candidates. In order to do that, we've had to develop a GMP NanoAssemblr system. And that system can move into a clean room. All the components of the NanoAssemblr GMP system are traceable. They come with a full documentation package, including everything you need to operate, install, and for maintenance of the GMP NanoAssemblr system. Basically, to de-risk manufacturing and to consistently and reproducibly manufacture large batches of your particular nanoparticle to support late-stage preclinical development and also clinical development, clinical trials. So After this call, please feel free to reach out to myself or Padma, and we can provide more information on our GMP NanoAssemblr system. But you can be rest assured, if you are using our benchtop instrument, which is the Ignite, or our process scale-up equipment, the Blaze, you're going to get the same quality nanoparticles using the NanoAssemblr GMP system. So it's that seamless scale-up and very reproducible results. So importantly, you can actually do a lot of your process development at a much smaller scale, saving valuable raw materials, including, in many cases, expensive drug substance, especially if it's an RNA-based drug substance, by doing those important process development experiments at small scale, and then getting very predictable results as you go to the GMP NanoAssemblr system, and really reducing the amount of work that you have to do at large scale, because the small scale experiments are so predictive.
Lloyd, could you tell us a little bit about, you are part of our clinical solutions team that obviously engages Sirnaomics. Could you tell us a little bit about the stage at which you like to engage with a customer's project? When should they be reaching out to discuss their needs to move as quickly as possible?
As early as possible, actually, because I know a lot of people want to move their project forward quickly, and they may only have a concept. They might be very early stage, but as planning for success and planning to get into the clinic with your promising therapeutic or nanomedicine therapeutic, I think engaging us initially and seeing how you can take your formulation a very early stage and how you're going to move it through preclinical development and get it to the point where you can have submit a clinical trial application or an investigational new drug application, an IND. I think that's really important. I think so. That's typically what happens, where we'll engage with a client or an organization that wants to move to the clinic, typically within a 12 to 18 month time frame, and we'll have an initial discussion with myself or someone in the clinical solutions team, and then the conversation will probably focus early stage on formulation and process development, and other members of PNI's contract services team are available for those early stage discussions. And the clinical solutions team really gets heavily involved once a given nanomedicine formulation is the process and the formulation, the process and the actual formulation itself are locked down. So that's when the scale-up work takes place, the more formal process studies, and manufacturing batches for larger preclinical studies, for stability studies, and also to basically prepare for a technology transfer, a tech transfer to a facility that can manufacture GMP nanomedicines. So PNI can help our clients and partner organizations to basically help move the project forward as quickly as possible, but not compromising quality in any way. I think that's we do consider quality as kind of the underpinning of everything we do here at PNI, and allowing clients to really understand our manufacturing process. It's pretty open. We're more than willing to help in any aspect of development. There's no black boxes, so to speak. This technology is readily available, working with PNI, of course. Also, I think importantly, technology transfer, and this is a little bit of what David will have talked about if he gets to finish his presentation today, and it's captured in the slides that I think will be shared after the call. Technology transfer it's a bit of a leap, because it really takes what you're doing at your facility and moving it to a facility that is focused and basically specialized to manufacture under GMP or good manufacturing practices to support, again, a clinical trial application, and to initiate, using the material you make at this GMP facility, initiate clinical studies in human beings. So It's a big jump for tech transfers, and that's something else that PNI is able to help with, and actually was able to fully support Sirnaomics in enabling transferring the manufacturing process from a lab environment to a manufacturing facility selected by Sirnaomics. That was intense work, and it involved transferring not only the GMP manufacturing equipment, but also all of the processes and all of the traceability from all the components that see the drug product. So anything fluid contacting, we have full traceability. We have through our quality group and our quality management system. So, rest assured, for tech transfer activities, we've got you covered.
Great. Next question we have, how to sterilize and remove cryogen in the system, whether the system can use single-use pump and tubing and chip, and can the software do audit tracking?
So, yeah, so for the GMP system, the entire fluid contacting path, the fluid contact path, is single-use and fully disposable. So for the tubing, that can be autoclaved. The microfluidic mixer itself, or the mixer assembly, is provided double bagged and gamma irradiated. Typically for nanomedicine manufacturing, sterility is not a requirement at the particle formation step. So we typically have a sterile filtration step at the end. Most of the nanoparticles that are manufactured typically are between, I'd say, 60 and 150 nanometers in diameter. So they can be sterile filtered through a 0.2 micron filter. Additionally, for the software, the current configuration of the GMP software is that it's the software is actually, there is not a data logging with the software. So for the current, this iteration of the GMP system, the values are manually recorded in a batch record.
Great. Next question we have is, do you support formulation development? How long does the process take? How fast can I expect results?
You can expect results fast because we're quite responsive once we receive a formulation development project. It really depends on what it is, what we are developing. Typically, nanoparticle formulations are complex formulations, so there would be multiple components that make up the nanoparticle. There are considerations to the buffers that you select and choose to get the most desirable particle. But the easier part is that our microfluidics technology, which allows these production reproducibly very quickly of high quality nanoparticles. So, I can't give you a precise number, but if you had a lipid nanoparticle formulation with a payload, a nucleic acid payload, typically after starting work, we'll have results within 1 to 2 weeks, initial results that we're able to share with our clients. And we're very responsive, looking at weekly calls to share progress, and we share all the data that's generated. So it's, I think people are pretty happy with how quickly we're going. Plus our experience, if you add up all the experience within our formulation development team, it's well over 100 person years of experience. And this allows us to kind of identify challenges and provide potential solutions for clients very quickly. So really, we do, we dip into our experience quite a lot, in addition to our capabilities as well. If a custom solution is required, that's also possible. And what I mean by a custom solution is if someone has a special challenge and needs additional functionality to be added to their kind of particle formation, we can do that by customizing or configuring at least our GMP NanoAssemblr system, and we can do some of that work with our other systems as well, if required. So, that's important to keep in mind as well. And that was go ahead, Tim.
No, just go ahead, Lloyd. Sorry, I thought you were done there.
Yeah, and that's certainly the case for Sirnaomics, where we needed to kind of think a little bit outside the box, but we could use our our base GMP system and modify or configure it for the client's specific needs and requirements.
And I was just going to add there that when it comes to formulation development and optimization, there's a lot of options you can pursue that our team can pursue with you. If you're coming with a blank slate but want to access, we do have delivery technologies, and you want to just access from a library that we've worked with, that can move very quickly. If you're coming with a new modality, such as the Sirnaomics proprietary platform, then our team will come up with a plan to very quickly characterize that and get you results. As Lloyd said, oftentimes initial results in a matter of weeks. But even with a proprietary platform like Sirnaomics as the story David will tell us at some point, will tell you, is that we moved from doing the first experiments on that to having them tech transferred in a matter of months. And so it's really, it can be very responsive, and the power of the platform really shows with its speed.
Yeah. So,
next, I don't know if you want to, I got another question, more questions for you. How does MW or quantity of DNA affect the formation of the nanoparticle, size, or zeta potential change?
Okay, I can help with that one. So actually, PNI has experience with various sizes of nucleic acid, whether it's siRNA, which I would say is probably on the smaller end of the size spectrum. Considerable experience formulating with siRNA with high encapsulation efficiencies and particle sizes that can be very small, starting around 50 nanometers, typically the 50 to 100 nanometer range ballpark. Recently, in the last few years, we've had more experience formulating larger payloads such as plasmid DNA, which is double-stranded closed circular DNA. And a lot of interest, obviously, in messenger RNA and also replicon RNA or self-amplifying RNA. And those payloads can get large. They typically, I think, around 1,000 base pairs, all the way up to greater than 10,000 bases. So it's we see encapsulation efficiencies typically above 95%. Even for these largest payloads, and particle sizes are, around the 100 nanometer range. So we can make a small particle that has is reasonably homogeneous, has a low polydispersity index, even for these largest payloads, using the same technology, using microfluidic mixers and similar buffers. Some of the buffers are different and tweaked to maximize encapsulation efficiency. As far as zeta potential goes, that's a measure of surface charge. And that really is dependent upon not only your negatively charged nucleic acid, but the other components of your formulation. So if you have any kind of surfactants in there, the molar ratios of each of the lipid components. So it really depends upon that. So it's not just the nucleic acid that determines the zeta potential of the resultant particle. So that typically has to be determined experimentally. And really, if zeta potential or high zeta potential is a requirement for your formulation, that would be part of our formulation development work that we could perform with the client.
Okay, the next one is, can you name the CMOs or any CMOs that you have experience using your GMP equipment?
We can provide potentially that information offline. I don't really want to get into the details of recommending CMOs. We work with a lot of CMOs. We have ongoing discussions with most of the CMOs, if not all of them, that work with these types of nanomedicines.
I think Lloyd, as are featured on our website, we can mention we have manufacturing collaboration agreements with Evonik, who is based in Burnaby, with Agi Biopharma, who is based in San Diego, and Fujifilm, who is in Japan. However, PNI, our model is a flexible model, and we work with whatever arrangement works for you. So, as our customer, if you prefer your own CDMO, then we can certainly do a tech transfer there. If you prefer the technology transfer to be into your own GMP manufacturing facilities, as a couple of our clients are planning right now, we can do that. Or we can recommend, our own CMO. And we continue to expand our CMO network. So we are currently working on CMOs in Europe and other parts of the globe.
Yeah, so obviously if we have an existing manufacturing agreement with the CMO, things will move more quickly because a lot of the agreements are in place, the technologies in place at that particular CMO. So, your lead time or setup time is going to be reduced.
I think that's, again, we ran over time, but, thank you for David. So we were able to finish the presentation. So I think we would like to log it off, and then we invite you for our next webinar on June 25th by Dr. Marcel Bailey, again, on the topic of challenges related to lipid nanomedicines.